This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure with the Convergent Science Network podcast with my colleague Tony Prescott. And we're here today with Neil Burgess, who is a speaker in our 10th edition BCBT Summer School. And Neil was giving an overview of his work on spatial cognition, also about how to link neural dynamics with advanced psychological function, like our knowledge of space. And Neil, what you also presented very much in your description of your work is this close coupling between theoretical approaches, modeling work, and experimental work, right? So where did this combination of these two methods, if you want, originate?
Well, I was originally a computational person. Having done theoretical physics, I started to want to do computational models of memory and other forms of cognition at the level of neurons. And my PhD supervisor, theoretical physicist called Mike Moore, said, if you're going in this direction, it's an experimental subject and you need to get involved in the experiments. And I'm always grateful for that advice. There would be no point sticking to, say, mathematically tractable models if they had nothing to do with the biological reality. And so it seems very important to have your models directly address experiment, and equally for your experiments to be, theoretically well grounded to answer particular hypotheses that make sense. And so, they experiment and theory should always work together.
Mm-hmm. But now, how many of the models you have generated in that period have you actually completely rejected and changed your mind about and taken a completely different direction?
That's a good question. I think that typically you're interested in a phenomenon and you continue to follow that even when experiments or models don't work. And so if you're interested in explaining that phenomenon, your models might change, and indeed your experiments might. So in terms of all-out rejection, I'm struggling. So I started off doing kind of Hopfield model type models of memory. And I was working with the psychologist Graham Hitch, who was interested in memory for serial order. And it seemed that although you can remember sequences of things in Hopfield models by associating one pattern of activity to the next, in human working memory for serial order, often you make paired transpositions of items. So instead of A, B, C, D, you remember, or you output A, C, B, D. And it's very hard to get that kind of error in a straightforward Hopfield model. And so I started using competitive queuing type models. And I haven't really looked back on the Hopfield type models, although I still believe that the attractor network idea is key to how much of the brain works, including hippocampal area CA3. I haven't really gone back to those models as just a pure attractor model of memory.
Mm-hmm. Right. So then you started out by saying, by pointing to this Wang and Simmons experiment that makes the point that actually there are a number of cues or a number of streams of information that come together in our understanding of space. So why do you think that experiment is sort of critical as a starting point of the analysis?
Well, it's just one experiment. I don't think it's critical, but it very nicely presents the fact that you can remember where something is in many different ways. And so it's sometimes good to start with that example, so that even though I then might drone on about hippocampal place cells for a long time, the audience already knows that that's only one kind of strategy for remembering where something is, and that other parts of the brain would be doing equally useful things that can help you remember stuff in different ways.
Mm-hmm. So of these different possible ways to deal with space, as a concept, how would you then define the way the hippocampus solves the problem of space?
Well, I mean, all of that part of the model is very much driven by the place cell phenomenon and surprisingly, single neurons encoding for single places. I mean, it's a remarkable sort of one-to-one mapping, aside from the fact that different cells will fire in different environments, and provides a very easy-to-use code. You could associate things that happen in places with the activity of the place cell that fires in that place very easily, or you could, having found a place cell that fires in a given place, it could be that its firing rate is modulated by some other factor that happens in that place. And that's just a very simple starting point for thinking about a memory system for location.
Mm-hmm.
Yeah, I think going back to this question about parallel representations, a lot of the hippocampal models, and I think yours, start from this assumption that a hippocampus represents one solution to the question of where something is, but an alternative Bayesian view might be you'd represent a probability distribution across multiple solutions. I mean, is that something that you entertain as a possibility?
Yes. So what's been lacking is direct experimental evidence that you might be weighing up the different alternatives and seeing, place cells firing for both different alternatives. But in many situations, you can see, for example, as I showed, the single firing field of a place cell becoming bimodal when you stretch the environment, for example. And it's possible that you could think of that as part of a system that's trying to estimate location. And because it's been predominantly taking evidence from distances to boundaries, it's now got a hypothesis that is, has two peaks in it, and that you are indeed representing a distribution. And in the experiment that I showed, where people had to remember where something was, the population vector overlap with the stored place cell representation captures well the distribution of responses across subjects in that experiment. Rather than indicating a single location. So,
yeah, but I think Tony's question is also annoying, right? Because in some sense, Tony is saying sort of this spatial cognition can happen in many different ways, in many different areas of the brain. It becomes a little bit like wishy-washy, like, okay, it's sort of not such a unique feature then of a single structure like the hippocampus. That's how I take that question. If it's just Bayesian, because okay, we integrate multiple factors, and so then why worry about the hippocampus? So, but I still believe that hippocampus is making a unique contribution to solving that problem.
Well, I would agree with that, but the question is that, I mean, so I think it's definitely true that we have at least two solutions.
No, but I'm here to make
yes, I realize
Neil's
that,
life difficult.
But just to clarify my position, so
okay.
there's a striatal solution to the navigation problem. There's something in the hippocampus. There may well be other ways of navigating, certainly towards sort of nearby targets, sort of, and so on. And so there's you can almost say, well, in the hippocampus we might be representing multiple solutions, and then the decision gets made in some other part of the brain about which way we would go. And there are models of hippocampus that are consistent with the idea of representing multiple hypotheses. So.
No, but I want Neil to tell me now which one it is.
Yes.
Well, I mean, as I showed, there are some things that we do know about the brain, and this Packard and Magor experiment was, again, it was only a summary of lots of things that people knew up to that point, but it was a very nice, clear expression of the fact that different solutions could seem to be supported by different parts of the brain. And you could actually inject anesthetic into the two different parts of the brain and see the different strategies being expressed in behavior. So in answer to Tony's question, I think, well, and indeed yours, that if I wanted to characterize the hippocampal contribution to these kind of spatial memory tasks, it seems to be representing a location relative to the environment, probably as a conjunction of many kinds of cues, some of which are distances to boundaries in different directions. Whereas other parts of the brain might be remembering more, I turned left at the shoe shop, or a sequence of turns on a well-known route. Or in other parts of the brain, still, a visual snapshot matching. This looks like where I was before. And all of these things are happening in parallel. And it is an interesting question. How they get combined, who chooses what. And indeed, on the input, there's different kinds of evidence coming in, and they are probably weighted in a Bayesian way with more things that seem to be more stable over time or more certain being given a stronger weight than things that seem more variable.
But would it be fair then to say that the hippocampus really is critical in generating this allocentric representation of space?
Yes, I would think so, yeah. But in many of these experiments, they're a memory experiment. You have to remember a location and go there. And we know that the hippocampus is critical for many forms of memory. And so in a spatial memory experiment, yes, if you inactivate it, then often the person or animal will not perform very well. So there's good evidence that the hippocampus is responsible. And in that Packard-Magor experiment, you could see that it was particularly responsible for one of the two available strategies for that task. Yeah.
But the general hypothesis that you're following then is that there's a population response in hippocampus of place cells, which in some way represent the brain's best guess as to where it is in the world in an allocentric coordinate frame.
In those tasks, yes.
Yeah.
Okay, but then, so now we have an hypothesis about what the hippocampus is doing, and it gives you this an allocentric representation of space, so in world coordinates, which is a fantastic accomplishment. And now we have to try to understand how this is done, right? And then you build very much your theory, your model on that around, at least in your presentation now, around this notion of the boundary vector cells. This is really the starting point of your whole analysis, right? So why do you feel that this notion of boundary vector cells is then so decisive to understand this ability to generate allocentric representation of space?
Well, first of all, the head direction cells, I think, are fundamental because this overall orientation, sense of orientation, is important for everything that comes subsequent to that. But taking that as granted, then you need some distances from some things to know where you are. And really, I think that if you were to imagine an animal with a sort of rangefinder looking at the distances to stuff around it in full 360 degrees, obviously the biggest contribution will be to either things that are very nearby or things that are quite extended. And so topographical features of the environment that are extended are going to dominate that kind of representation. And I think that that's really the boundary vector cells. We heard also, and I talked about object vector cells, and we heard from them, from Edvard Moser. The boundary vector cells do actually respond to small objects, just they have a very small firing field is provoked by a very small object. And so they're sort of responding like a rangefinder tuned in a particular direction, allocentric direction, presumably governed by the head direction cells. And once you're oriented, the next thing you need is distance. And these are rangefinders tuned to directions, and they seem to provide the simplest explanation for the sort of broad qualities of the firing fields of place cells when you put an animal in different environments that differ in shape and size, but not differ in texture and odor and so on, enough to make different place cells fire, in which case you wouldn't be able to try and analyze these different kinds of inputs.
So then you're saying, look, I have a heading vector. This gives me, if you want, that's my compass, that's my directional system. And now I glue to that some sensory feature. Might be somatosensory, it might be auditory, it might be visual. And that now gives me, let's say, a reference in space of how that compass coordinate maps onto that specific environment.
Well, I should say that in most normal environments, there would be a broad range of different local cues also, which would help you in a very direct way to localize yourself. And they are probably also important to place cells. But in most of the experiments most people do, they try to control for those cues so that they're only the cues that they're aware of remaining, like the boundary of the environment, these distant orientation cues for the head direction cells. And so given that you've tried to remove reliable local cues, also usually the floor is rotated between trials, then what do you have left? You have distances to extended things.
Right. But now the head direction system depends on path integration. Path integration is noisy because it's an integrator. So isn't that a little bit of an obstacle for such a system to keep on working reliably over extended periods of time?
Yes, indeed. So that's an interesting thing that I didn't talk about, but the same, exactly the same concerns about how you translate between egocentric and allocentric in terms of place cells and sensory inputs, which have to be egocentric, applies to head direction cells. So a head direction cell will fire whenever the animal's facing, let's say, north. I don't actually mean compass north, wherever it is in the environment. And so actually in all experimental situations when they've been recorded, that tuning is parallel across the environment. But if they were simply responding to sensory cues, you would see parallax, which you do not see. It is an allocentric response, the head direction cells. And so again, you need to have this translation mechanism between egocentric sensory input and this allocentric response. And so we think probably retrosplenial cortex, as in the model that I explained, has to do that same job for the head direction cells so that they can be reliably anchored to sensory input to prevent the accumulation of error that you would inevitably get if you're just integrating angular acceleration.
Right. But then is the sensory cue that you link the heading vector to, that in itself might already be an invariant representation of some sensory state. It might not be necessarily egocentric, right? If we go through some perceptual hierarchy, and if I climb up that hierarchy, I might get to more invariant representations of space. Maybe I have a very abstract representation of, say, just, okay, this is a wall. I'm not interested anymore in its orientation or its color or its size, a wall. So how would that notion of invariant representations of the sensory feature then actually be a challenge to that model?
Well, I think it's the invariance we're talking about is exactly, the head direction cells show that very nicely. They're at the top of the hierarchy for direction. They're invariant to all of the parallax and sensory stuff that happens as you move around, and they extract what would be a compass, and the rest of the system is probably built on that.
And now, if I would replace the heading vector with a movement vector, would that change your approach very much? Because
I'm not sure what you mean.
As opposed to integrating, let's say, my rotations in space, I could also say I just interpolate between subsequent movements, and this gives me a vector that I might then use to get to some sort of allocentric representation of space.
Well, so there's two separate things here. One is how do the head direction cells fire to encode the orientation of the head. If you then move on to path integration and perhaps what drives grid cells to fire, then you have to integrate movement rather than head direction. So you need to know which direction you've moved in, not which direction your head is facing.
Exactly. So would it make a big difference to your proposal?
Not for the boundary vector cells, I don't think.
Because usually the head of the rat is
because the boundary vector cell model of place cell firing is not path integration. It's just feed forward sensory input. What direction from the head is the boundary feed forward? So there's not an issue there. When you come on to how you do path integration, how you integrate your own bodily movement to estimate where you are, which may well happen through grid cells, then indeed you need to know your movement direction, your movement vector, not your heading vector. Right. Perfect. Yeah. And so there's various hypotheses as to where this signal comes from, because it can't be just the head direction cells multiplied by running speed. It has to be movement direction.
Would that be your grid cells then?
No, what we're talking about are what are the inputs that would go to something like grid cells that is the movement signal that is integrated. Yeah.
No, but wait, if I would just look at subsequent grid cell responses, I could infer a movement vector.
You could do.
So that's what I was after.
Yeah, so I think maybe this question came up with Edvard Moser's talk, that whether you could look at it either way. If you think grid cells know how to fire because they're doing path integration, you need an input, which is a movement vector. Or you could say, well, grid cells fire as they do, in which case a movement vector could be an output. Yes.
That's it.
Yes,
exactly.
Indeed, it could be. But then you're still left with the question of how the grid cells fired in that way in the first place.
Right.
So the activation of the place cells you have being driven by the boundary vector cells, and so it's basically a thresholded sum of the inputs from the boundary cells. clearly you could choose the weighted sum of the inputs as just the kind of the first thing to try. Are there other motivations for doing it that way? For example, from sort of environment warping experiments that say that it's the weighted average, it's the best thing to use?
Well, it was the simplest thing first thing to try, but in some experiments, as you expand the box, you have a place field which slowly reduces in firing rate and disappears.
Right.
Which implies a sum with a threshold. As the initially overlapping inputs from opposing walls move apart, then the bit you've got left slowly falls below threshold, and the cell stops firing. So there's good reason to use a thresholded sum. There's a reason for a threshold, and we really did see in some cases individual sub-peaks being moved apart, which means you have to sum the two rather than multiply. If you multiply them, then you get, for example, a Bayesian, average position in between the two peaks as they will.
Well, this is
apart.
what I was getting at. Is there support for multiple hypotheses in some of those experiments?
Just to finish answering your question, there's clearly adding these inputs from the different walls and having a threshold was the most obvious thing to do, given all the things we've said. So if you were trying to combine hypotheses probabilistically, you would probably multiply. That's not what we saw.
Right.
It's interesting because of these experiments by Galastel and Cheng, and also by Cartwright and Collett. John O'Keefe, in this original stretchy box experiment, was expecting to see place cells firing in the constant ratio of the distances across the box, rather than the sub-peaks being pulled apart and sticking to the fixed absolute distance from the walls as they were pulled apart. And so if what he'd been expecting had happened, that would have been an argument for multiplying and having kind of the combined estimate of two probability distributions, but that isn't what we saw.
But it sounds like what the experiments say is a bit more sophisticated than the weighted sum, than the averaging. But so there might be something to look at there in the future in terms of how in this inconsistent environment that's, where the boundary vector cells don't all point to the same place in space, then you might start to represent multiple hypotheses in terms of place cell activity.
Well, I don't know if there's evidence for that. I think there is definitely evidence for hypotheses about what happens in a place. A place cell might fire in the same place trial after trial, but its firing rate might vary quite strongly according to other hypotheses to do with sensory stimuli that are present, whether objects are nearby, smells, how fast you're running. So I think there's a lot of orthogonal hypotheses to place, which also modulate the firing rate, but it more like a gain field representation.
Okay.
So the actual firing field itself isn't obviously doing something nice and Bayesian about estimating location. When you do these weird experiments, when you deform the environment, which are rather unnatural, but they probably are combining the cues to them, such as we haven't talked about path integration input and the sensory input are probably being combined in a more Bayesian way.
Yeah, and also, as you say, maybe there's something happening in time, that you can swap between hypotheses over time and explore different.
Well, actually, on that point, if you do this stretching experiment and you get a single place field stretching into two place fields as you've expanded the box, then you notice that each place field fires a bit more according to running direction. So the place field that appears to be attached to the wall behind the animal fires a bit more, so that when you're running in one direction, one peak will be higher, and when you're running in the other direction, the other peak will be higher, which we interpreted as being this path integration input, adding to this environmental sensory input, and being stronger if you've just run from a wall. Obviously, you have a good estimate of how far away it is from path integration, so that peak gets a bit higher because it's got a stronger path integration input than the peak that's attached to the opposite wall that you're running towards.
Yeah.
But I'm find it very interesting that you guys seem so, let's say, motivated to interpret this in Bayesian terms. Well, if you look at the dynamics of CA3, CA1, where the real action in the end happens, it's also highly competitive, and in the end, it's relatively sparse. You can also think about it much more as, let's say, the endpoint selector that sits on top of some Bayesian integrator, as opposed to being a Bayesian integrator itself, because you just don't have the dynamics for it, because it's rather sort of selective, rather sparse, and these are not the features you want in your Bayesian integrator. So is that not fair to place that bit outside of the hippocampus, gentlemen?
No, I think that's fine. As I said initially to Tony, there isn't good evidence of multiple hypotheses being entertained at the same time. It would be a good way for the system to work, but it may well not do that.
And then can't we not explain the elongation of the place field also in terms of a perceptual learning process? Because the animal starts to also carve out specific trajectories through that space. It's just not moving randomly, right? And the cells you measure from is probably also on trajectories the animal will visit more frequently than other trajectories. So it will actually be moving more often from that specific initial position to a final position, and therefore expose itself more to the specific sensory features that are already associated with that place field, and therefore strengthen the specific intra-place field associations, and then, if you want, elongating its response.
Well, there are interesting trajectory effects on place cell firing, but they're rather different than these effects in this unusual situation where we change the shape and size of the environment. I mean, I think that's an unusual experimental manipulation that shows us something about the inputs. However, the trajectory effects are interesting, so that if the animal's always running in the same direction, you see that firing fields tend to elongate backwards a little bit along the track. So Mayank Mehta noticed this. But that's been interpreted as perhaps the place cell beginning to fire in expectation of getting to its, quotes, true place or original place. And there's some interesting theoretical work by Matt Botvinnik and some co-authors suggesting that these place cells might encode a successor representation for reinforcement learning, which is an idea from Peter Dayan in the 90s, that if you were to tweak your state representation to actually include the likelihood of eventually ending up at that state, even when you're at other states, then that enables you to estimate future reward at a given location much more efficiently. And so it's possible that if you have repeated trajectories, then you can build a little bit of the probability that you now know that if you're here, you're likely to end up somewhere else into the state representation, so that you can then rather more easily estimate likely future reward from a given location, because you've built in the transition probabilities, which are now not uniform because I always run in this direction when I'm in this place.
So it's the kind of model that reduces the whole brain into the hippocampus.
Well, no, I mean, it may be that that kind of representation, perceptual learning and so on affects, it could explain what happens in lots of parts of the brain, not just the hippocampus.
Right, okay. But now, so in your physiology, so then with that model, you went to look at the physiology, and then you show that these sort of vector border cells are to be found both in entorhinal cortex, or subiculum and entorhinal cortex, which is interesting, right? Because if you look at the overall loop, we start in entorhinal, we go to the dentate, gyrus, CA3, CA2, CA1, then subiculum, and then we go out to entorhinal cortex. So isn't it really strange that we find these cells actually at the input stage and the output stage of that whole loop? It's like we're wasting all this circuitry in between to do nothing, and we just have our border vector cells.
It is strange. I mean, it is a loop, and we may be interpreting how it works incorrectly when we think of entorhinal as the input and subiculum as the output. I mean, there's plenty of connections from subiculum back to entorhinal, and pre-subiculum has very strong connections to entorhinal. My colleague Colin Lever is always keen to point out that something that Pat Sharp originally noticed, that when you get a complete remapping of place cells in CA1 between two environments, there's actually spatial responses in subiculum that are nice and stable that you can record, and they show no remapping at all. And yet the standard model would be that the major input to subiculum is the output of CA1, and yet you've got this complete change in CA1 representation, no change in subiculum. So yes, it is a puzzle. Maybe subiculum is the input, and it goes around to entorhinal and then into, who knows? But it is a puzzle. It's puzzling from the anatomy. You wouldn't expect it.
No, but it's quite a challenge
there's something there that...
but it is a challenge to your model, right? Because you want to say these border vector cells are, if you want, a representational primitive on which I build everything else. Yes. But now we see wherever you want to put the input and the output, they still sit there.
Well, and what is also true, if you could also ask me the sort of functional difficult question would be, the place cells are a basis set. It could be that boundary vector cell firing responses are made out of weighted sums of place cell firing. That could be possible.
But now, if you look at subiculum and entorhinal, how big a proportion of cells in those areas would actually show these properties?
So I think it's around 20% or so is Colin Lever's best estimate in subiculum, and it's a bit less, maybe more like 10%, but still quite common, these border cells that Edvard Moser described, i.e. the boundary vector cells that fire quite close to the boundary. They're quite common. There have been a few reported that fire at a distance, but it's a handful. And so that's another strange... that remains to be the exact relationship between the border cells and the boundary vector cells and the object vector cells. It is still not quite clear.
Okay, so but then could we argue that in entorhinal cortex there's, if you want, a rank order of cells, and in terms of how foundational they are? You could argue, well, grid cells might be very foundational. Single modality cells in lateral entorhinal cortex might be foundational. And now I start to merge them. I start to merge them in your border vector cells. So because now I have, within entorhinal cortex, I have access to both movement vectors, grid cells, and have access to sensory features of the world, lateral entorhinal cortex. And I just have a subpopulation of cells that is associating the two, and now I have my border vector cells.
Yes, the grid cells are also a basis set, and you could form any responses out of them. So it's a good question, although it's not quite clear what foundational means in your question. And so we have looked developmentally. So Francesca Kikuchi in John O'Keefe's lab was looking at this, and also Edvard Moser's lab at the same time. And what you see during development as pups learn to start to crawl around and open their eyes and so on is that the head direction cells are foundational in the sense that they seem to be there as soon as you can record them. There's also theta rhythmicity as early as you can record them in this situation. Then you also see place cell responses as they start to crawl around, the rat pups. And they get better with experience over several weeks. So there's a slow improvement in the spatial tuning of the place cells. Grid cells are not present until 4 or 5 days, developmental days after you see these other cells. And so they're not foundational in that sense, it seems. And this has led to people suggesting that maybe the grid cells require some stable spatial input, maybe from place cells, to wire themselves up to fire properly. I mean, the head direction cells seems to be a very strong attractor network, so that the head direction cell responses are mutually coherent with each other, even if they are drifting a little bit early on in development. And it may be the same with the grid cells, that they're mutually connected well, so that it's an attractor, but it's just drifting so much relative to the world that you can't ever record it. That's a possibility, and then you only see it when it gets attached to stable sensory inputs, because then you can average over movements across the environment. But yeah, they're all interesting questions, and you can try. The good thing about this field is that you can try to answer them with experiments.
Absolutely.
And the developmental trajectory you're talking about there is based on the chemical properties of the cells, not their functional properties.
No. Sorry, the function. This is recording the spiking activity as pups first start to move around, as they start to explore around the nest. Francesca will give a talk about this tomorrow, I think.
Right. But now the object vector cells, so this is the next complexification that also Edward talked about yesterday. You see them as a further variation on that same theme, or should we now be really shocked and say, oh, this is again completely different? And
well, they're obviously closely related to boundary vector cells in the sense that firing at a allocentric displacement vector from something. And the only thing that's different is that they seem to be specifically tuned to small objects.
Right.
So a boundary vector cell, as I said, will fire to an extended object, but will also fire to a smaller object but just with a very small firing field, because obviously it only has to move a little way, and then it's not in the receptive field for that cell.
Right.
Whereas an extended boundary will produce a long strip of fire. The object vector cells seem to be different in that they don't seem to respond to the extended boundaries of the environment. They just respond to objects put within it. Now, it could be that over time, there's something to do with the object being novel and the boundaries being familiar, means that it discriminates, because the same object vector cell will respond similarly to different objects. It's not specific, it seems, to specific objects, but will fire to any small object, but not to an extended boundary. But so far, the experiments I don't think have really been done to see whether if you put a novel extended object in that looks a bit like a boundary, whether those cells will fire or not.
Exactly,
that
that's the
hasn't
question,
been done.
right? When does an object become a boundary?
The reverse experiment has sort of been done in that Bruno Pousset's group had showed that some wine bottles put inside one of these boxes when you're recording place cells typically don't cause much place cell firing, although we know now from Jim Neerham's work that a small number of place cells will fire relative to these, indeed, looking like object vector cells. But when they placed 3 wine bottles in a row together, they started to have more influence, probably because of an extended thing, they would be driving more boundary vector cells. But the same kind of experiment hasn't been done with these object vector cells yet.
But the interesting thing about now these object vector cells is that they appear in CA1, apparently. No?
Well, a very small proportion of CA1 cells, yes. But Edvard was reporting a medial entorhinal and some interesting sort of memory times object cells had been reported in lateral entorhinal too.
But can't we? But look, these are highly plastic circuits, and they are exposed to multiple, let's say, streams of modalities and submodalities. So if we just combine these two considerations, is it then not sort of predictable that these kinds of combinatorial or conjunctive encodings emerge rather naturally?
Well, yes, but before we proposed them from our analysis of place cell firing, nobody knew that an offset vector, an allocentric offset vector, was one of the ingredients that would get combined with boundary detectors or whatever. And until the Mosers recorded grid cells, nobody thought that this sort of Fourier-like representation of space would exist or needed to exist. And so, yes, in retrospect, but before the fact. No,
I didn't want to trivialize it. I just want to say this might mean that if you now go for some ambiguous border object, for sure you'll find a cell that will respond to it, because we look at this combinatorics.
Maybe, but I mean, the interesting thing, I think, as Edvard pointed out about the hippocampus and entorhinal cortex, is that coming from the point of view of a Hopfield model for memory, you might expect a random distributed binary code that was uninterpretable. And in fact, you see these incredibly clear, discrete responses, say to compass direction or to location, or, a Fourier-like grid, or, a boundary vector. Now, there are, as Edvard said, you do get conjunctive directional grid cells, for example, and there may be other conjunctions, but the overall impression is that, and there are plenty of spatially modulated cells that haven't been characterized, also in entorhinal cortex. They're probably now less than, they're probably a minority of all the cells, because most of the other, because so many cells have been characterized. But still, the overriding impression is of these incredibly discrete types of encoding being present. And sure, they are present in some combinations also, but overall, you wouldn't say this is just a mush of everything. You would say, Jesus Christ, why are those such specific responses there?
But as a mathematician, it must appeal to you to find some theory that explains the emergence of these different kinds of cell types, which are particularly useful for navigation, and then to be able to perhaps generalize that and say what further kinds of cell types we might be able to predict that we should see, perhaps because they're adding more orthogonal information to the mix, perhaps sensory cells, for example. And I think that one of the things you alluded to, and also Edvard in his talk, was how the grid cells have this sort of change in scale, which follows a sort of mathematical law, which is consistent with efficient coarse coding. So it looks as though there is again, something which doesn't look at all accidental, but must be the result of this system responding in some very effective way to choose the right inputs for mapping space.
Yes, I think when you come to the grid cells and the discrete jumps in scale, that is clearly well arranged for large-scale navigation. You get a big combinatorial power for the range over which you can encode locations with that kind of representation.
So what is your prediction for the next kind of cell type?
It's funny. I don't think the field at the moment is working that way. It's true that we predicted boundary vector cells from looking at how place cells responded, and going back, that argument, that way of thinking, was certainly used by O'Keefe and Nadell. And John O'Keefe, having found place cells, predicted there should be directional cells and something like path integration or distance cells. And so early on, having found place cells, he did predict the head direction cells and then something to do with distance. And they have been borne out. And something to do with vectors, it seems like, is necessary. And we have boundary vector cells and object vector cells now. I think the interesting thing, so there's already a lot of ingredients there for making quite a sophisticated system for space. And at the moment, I think people are thinking more, how could this, all this stuff we've learned about space tell us about more general memory properties of the hippocampal system? That, how does it store other information, which we know that the human hippocampus is required for all sorts of forms of memory. And I think that's interesting. So I haven't predicted any more kinds of spatial cells, I must say. But
that's particularly what I was getting at, sort of human episodic memory, which is more than, about a lot of things, more than space. And where it's less easy sort of coming at it from the geometry or whatever to say what kind of basis functions you would want to have. So we might look at some mathematical way of finding appropriate basis functions for learning about episodes in time. So.
Yes. Well, so I think it's very interesting that the cells in the human hippocampus, most famously Jennifer Aniston type cells, are, they're a local tuning curve to a concept, in this case of Jennifer Aniston among all famous people or people you might have heard of. And, a cell will fire to that concept, whether it's a written name or however you bring it to mind. And other cells will fire for other concepts, be they spiders or a semantic, well-known landmarks or famous actors. And so you might indeed see the sort of locally tuned place cell response as very similar to these locally tuned responses in semantic space. And this very interesting stretchy bird experiment by Tim Behrens and his group, showing what looked like grid-like responses in humans to semantics two-dimensional spaces, and in rats to one-dimensional continuously varying tones, showing place cell-like responses from David Tank's group, do imply that these principles that have been easiest to spot in space and freely navigating animals might well apply to all sorts of other kinds of information and how it's organized and how it's retrieved and so on. And then that would obviously be starting with these very well characterized spatial responses, I think is a good place to try and understand everything else, which is obviously much harder to get a grip on.
So that would suggest that one thing we might look for is sort of compression type algorithms, which will give us a low dimensional or a relatively low dimensional description of the current context, which we can encode then in entorhinal cortex and use to run our sort of location finder or memory finder algorithm.
Yeah, certainly
is that a good metaphor?
Well, the grid cells certainly appear to be a compressed code, like a Fourier code or something that would be very efficient compared to the place cells, which in their own way are very useful for tagging specific information to specific places, but are less efficient for covering large areas in a metric way where you can infer the vector between any two parts. If I have a place cell that fires in one place and another one that fires in another place with no overlap, it's hard to know how they're related to each other. Whereas with grid cells, you can infer the vector between two grid cell codes for two different locations. And so that's very powerful. It's a compressed representation. It looks a bit like a Fourier pattern, but equally, Jory Derdikman's group has pointed out that if you do PCA on the place cell input that you get as your animal is running around, then you get things that look like grid cells. So indeed, the grid cells might be doing a PCA of this place representation. And there's a similar argument to be made for the successor representation that I mentioned for place cells, in that the eigenvectors of the successor representation also look like grid cells. And so they're clearly related. The eigenvectors of the covariance matrix being the same as the PCA. It could well be that the grid cells are a compressed way of arbitrarily representing any of this location coded or, single peaked tuning curve, grandmother type cell response representation of arbitrary information, which is very useful for then associating things to that information in the hippocampus.
Yeah, so I think I maybe put words into your mouth, but you're agreeing that the entorhinal's a compressed representation of the context, and then that's expanded out in CA1, CA3, perhaps after being sparsified in dentate gyrus is often a step people add.
Well, David Marr's model of the hippocampus remains the best model of hippocampus in memory. It's probably surviving that, and his cerebellum model are surviving really well, maybe even better than his later work in vision. And yes, those ideas hold a lot of currency, although it's important to remember that entorhinal cortex is not just grid cells. So even within medial entorhinal cortex, it may be that head direction cells are more numerous than grid cells, and there are also these border cells and conjunctive cells and spatially modulated but not grid cells. So, yes, for the grid cells, they look like compressed code. There's other things going on, also sensory inputs to place cells, we think, not just the path integration type inputs.
But aren't you, first you guys are all swept away by Bayesian, and now you're all swept away by, let's say, compression, which in some sense is already happening to a large extent outside of the hippocampus. And earlier, I thought we had agreed that hippocampus is contributing to constructing allocentric representations. So let's not worry about compression then. But now you guys are all worried about compression. So what is it? Compression, allocentric, or is it both? What do you want?
Well, you're comparing apples and oranges. I don't. I think you can have an allocentric or an egocentric representation, and you might want it to be compressed or not. So I don't think we can compare allocentric, egocentric.
No, you have to. No. What I'm saying, compression or allocentric, is it both? Is it combined? Is one building on the other? Are these independent orthogonal interpretations?
Well. So those codes, the head direction cells and the place cells and grid cells, appear to be allocentric in the sense that the reference to the world. It's which way you're facing in the world, where you are in the world. But they are where the animal currently is, which you might say is egocentric. It's referring to the animal's own location or orientation. Having said that, I think then the grid cells look like a compressed version of the place cells. So it may be that an expanded representation is useful for attaching things to, and that might be the place cells. And a compressed representation might be useful for generating large-scale metrics within which to compare or know the relative positions of lots of information.
Now, about 10 years ago, we showed that grid cells actually carry more information about space than place cells. So if you do position reconstruction from grid cells versus place cells, you have more accuracy from the grid cells. So that would be then inconsistent with the idea that they compress the place cells, because then the place cells would have more information for position reconstruction.
Are you comparing the same number of place cells and grid cells?
No, this was low control.
Yeah, no, well, exactly. So if you have a sparse representation, which is easy to attach things to, then obviously it's more efficient. It is compressed. You can have a coverage of a larger environment with the same number of grid cells. That's why it's a compressed representation.
Okay, look, but now the other, so this is a controversy which we will solve later. But now you presented a model where you wanted to explain how now you could use allocentric representations to also reconstruct, if you want, imagined locations, right? How you could use imagery to sort of make now predictions that would be relevant for navigations. How What's the contribution of that model now to our understanding of the hippocampus?
Well, that model was trying to take these sort of spatial things that we recorded in and around the hippocampus and say, how could they apply to human episodic memory? And, the experience of human episodic memory is reliving the event. And in terms of the visual content, it's imagining the scene. And so the proposal was that this spatial system is a way of pulling out the stored allocentric or abstract information that you have in your temporal lobes to create a coherent spatial scene from a single location with a single direction, provided by the head direction cells and the place cells, respectively. And then how you could project that information into an egocentric frame so that you can imagine it. Mm-hmm. So that's a necessary thing that has to happen if we want to make contact with human imagery. And so that's why I put it in the model. In principle, you could solve vector navigation just using the grid and place cell model, and maybe you do that. You need this extra stuff to be able to sort of visualize. But of course, that gives extra functionality. You might, in principle, if you've got a lot of stored abstract information, you could pull it together to give you the viewpoint, if you like, on all of that information that's consistent with being in a single location. And that's a very powerful way of reducing the enormous amount of stored information. You're just going to pull out all those bits that are consistent with me being here, possibly facing this direction. Now, what is around me? And if you think conceptually, you've got a whole lot of stored information, a whole sort of life's worth of experience, if you like. If I ask you about Jennifer Aniston, you probably want to pull out just the information relative to that location in the conceptual space of all actors or people that you've ever seen. And so, it's more that these are ways in which memory could function. You have to consider the retrieval process. You could store an awful lot of information, but how do you retrieve it? And episodic memory seems to be a very specific way of retrieving information. You impose a particular location and perhaps a particular direction to interrogate your stored data with.
But now in the model itself gives you like a lookup table, right? You can sort of project back from the egocentric views to the allocentric views. And to what extent is the model then able to capture also the physiological and anatomical characteristics of this hippocampal system?
Well, it's quite a high-level model because it's trying to deal with, cognition, and we know so little that it would be inappropriate to try and build too much physiological and anatomical detail in from the start. But this translation circuit, as shown by Alex Pouget and various other people, corresponds to the observed parietal gain field neurons that have been recorded, and should function as a translation circuit, irrespective of what it is you're retrieving from allocentric or egocentric coordinates and translating into the other. I'm not sure if that answers your question, actually.
Well, because it's also a bit a step towards the experiments you did with human subjects, where you then start to match the response of the model to fMRI experiments with human subjects that were in some sort of navigational test. So how well was the match then between the fMRI results and the predictions of your model?
Well, in terms of the fMRI, it was more that we could have, to most of the areas of activity, that the areas in the brain that showed increased metabolic activity during this kind of task, we could ascribe some kind of putative function. It's very speculative, but the model says what that activity should be actually doing. In the hippocampus, the place cell should be doing this, and in retrospinal cortex, it should be a translation circuit or whatever. And so it's not really that there was a particular... The model wasn't good enough to constrain what activity you should see. It's more that my choices of where these bits of the model should be in the brain were broadly borne out, and they give us some kind of way of trying to understand what that activity might represent in terms of mechanism.
So should the model be as a heuristic to help you now to further think?
It helped to interpret those results. It wasn't that we predicted results and, we could measure the error between the predicted results and the actual result.
But now in terms of the performance, so you had humans in a virtual reality environment where they that to navigate around, there was a target that they had to sort of memorize.
So we did do a behavioral experiment where we did match the predictions of the model, behavioral predictions of the model, and the behavior of the people. And in that case, yes, as of the models we tried, the boundary vector cell place cell model was the best match to the distribution of responses across participants of where they thought this thing was when you change the shape and size of the virtual room.
Right. But we discussed it also during your talk. But this is, I think, an interesting challenge now, because you basically took the response of all these subjects. The subjects had to estimate where they were relative to this target. So this gives you distribution over all estimates over all subjects, right? And then you show that your model could capture well the distribution over that whole group. But now if we split it out to the individual level, and also you showed that data, you actually see that the individual distributions can have a rather different shape. Actually, it's clustered in, let's say, 3 different groups.
Much less variance within subject than between subjects.
So then, in the end, what are we modeling, right? Are we modeling statistically over populations, or do we have to take into account? Must we be able to also account for then these individual differences? Because you saw that the subjects that were accurate had a pretty nicely clustered set of responses.
And indeed, the ones who were inaccurate
were caught.
Well, to be fair, actually, in that experiment, because you've changed the environment, there's no right or wrong. In fact, you can say,
they had a task.
They had a task to put their marker where they thought the flag was.
That's it. Yeah.
But the encoding environment had changed.
Sure.
And so it's a bit of an arbitrary question, and they did their best. And some people, they made different guesses. But to answer your question about what the model was showing, I think the model was showing that as a population, people had a vague idea where things were that related to the walls of the environment. And the model captured some aspect of that, but didn't capture the individual differences by any means at all. But it was interesting that as a group, they kept within this sort of very broad distribution that the model could show. I mean, we could have cranked up the model and said, just sliced off the very top likelihood match for location. And that would have been, a very narrow distribution. And some subjects put their responses there and others put them miles away. And some subjects were also confused and did unacceptable things.
But looking at the distribution across the subjects, we see 3 groups, right? There's one group that say, let's call them accurate. And they're a bit at the northeast corner of the environment, close to the wall. The second cluster is sort of southeast, so close to the wall, but they're like reversed, right? They've mirrored, if you want that space. And the third group is completely off in the opposite corner. So is there a set of parameters in your model that would be able to then tune the model to capture each of those clusters of responses accurately?
Well, I think that there was one participant that was off in the corner. I think we have to just ignore. Who knows what they were thinking about? Then there's 2 other groups of responses, and indeed they're not. One is within. To see the blue one there is actually they've responded in both groups, but it seems like of these subjects, more of them, because the original location was nearer to the north wall than the south wall, more of them were, if you like, trying to replicate that distance to the south wall. Although they don't replicate that actual distance, it's stretched down somewhat. And then fewer of them, perhaps, if you like, paying more attention during encoding to the distance to the south wall, and they're replicating that to a greater extent. And so who knows, as I showed their viewing direction, it's not that the ones paying attention to the north wall were all looking at the north wall. Some of them were. But there's a few that were looking at the north wall that matched the distance from the south wall.
Sure. But it's interesting, because then the model replicated something that was not present in any of your subjects.
Well, we did discuss this during the talk. I think some of these subjects who are what you call quite accurate.
The blue one. Okay.
Yeah, well, and maybe this sort of reddish one that's in there as well. A few of them may have actually fitted it. But yes, the variance across subjects is not captured in the model. And it seems like you'd have to have different weights for the north boundary vector cell and the south boundary vector cell, and tweak that between subjects to get this sort of response in a way which wouldn't be very satisfactory. But you could do many, many trials. And then, see if you're still predicting their responses, if you could find some way of knowing which wall they're going to be paying a bigger weight to. We didn't try and do that. You might be able to. I don't
but do you see this as a critical challenge to the model, or you think that this is a detail that you can handle?
So that model only requires the boundary vector cells and the place cells. Mm-hmm. So now we have this more elaborated model, which includes the visual imagery, and would allow us to also incorporate what some of them seem to be trying to do by lining up their viewpoint at encoding and retrieval, which would be to match the current visual scene with the imagined visual scene from encoding. We could try to add that. That would be, of these three different frameworks that I mentioned at the beginning, I think the spatial updating or path integration probably can't be used here because there's a random teleportation to a different spot before they make their response. But the other two, the visual matching and the environmental location, surely are relevant. So we could try adding the visual matching to this and see if that improves. And maybe if something about the orientation at encoding gives some of this inter-subject variation, because their encoding positions are all different. That might be a way to go. We haven't tried to do that so far.
But that would mean that models on navigation would have to start to include a notion of individual style.
Only if you want. I mean, yes, if you're interested in those individual differences, or you might say, like we did in this original experiment, who cares? Let's see if we can get any kind of match to anything, averaged over whatever.
Sure, but now you have matched to an average person that doesn't exist.
Yeah.
It's a typical problem in modeling,
Indeed.
Right? So,
well, and other good points could be made, like all models are wrong.
Yeah, sure.
But actually, a model is only replaced by a better model also. And that, so.
Yeah, no, that absolutely right. So look, let's get to you then started to map these ideas, or also reasoning from these models. You looked at the response in the human brain using fMRI, but now the second problem that you could face there is, of course, also the matching in time, right? Because initially we talked about grid cells, place cells, so on, and we talked about electrophysiological data. This is all happening in milliseconds. And now we are validating the model against fMRI experiments where we actually look at a very different spatial temporal window of response. It's measured in seconds, right? So how do you overcome that challenge? That in some sense the dynamics of the model is completely outside of the window of the measurement technique that you use to validate the model.
Well, we made predictions that were appropriate for that kind of testing. We also use MEG and intracranial recording, which has higher temporal resolution, of course. But for predictions for fMRI, you need to, the most common kind of prediction is that on average during this task, this particular part of the brain is going to be a bit more active because there's neurons there that are doing something that are required for the task. And that's the kind of prediction that we made. So you may be referring to the sort of grid cell-like experiment where we tried to make a prediction for what would be the time-averaged bold signal as a function of running direction over the whole trial, by noticing that the grid cell firing patterns tend to be aligned across the whole population of grid cells. We tried to make a prediction for a difference according to alignment with the grid axes or misalignment with the grid axes in terms of your running direction, which we could look for on this, time average data over the whole trial.
Right. But that was a brilliant idea. How did you stumble into that? Or was really like a sudden insight, like, okay, the orientation of the grids has a certain discretization, and given that discretization, we must see alignment or misalignment, and we can exploit that in our bold signal? Was it really like...
Well, I should say, so the other two authors who are both postdocs in my group at the time, Christian Doehler and Caswell Barry, had been wanting... So Caswell Barry was recording grid cells in rodents, and Christian Doehler was doing fMRI, and he really wanted, the two of them really wanted to be able to see something in fMRI that might somehow relate to grid cells. And so we did discuss it for some years before, and Caswell noticed that these grid cells were aligned, the grid patterns were aligned, and so that we tried to look, and eventually I had heard of quadrature filters, which are a good way of looking for a particular phase orientations, and so we put this method together. And eventually applied it to data that we'd actually recorded over the previous 4 or 5 years for other reasons prior to coming up with this particular idea for looking for a grid-like signal.
Right. No, it's an amazing result. But now is the 60 degrees, is that, let's say, the optimal, if you want, misalignment between grid cells? Is that where you would find the strongest competition between their responses? Or do you see the 60 degrees as some sort of canonical feature of their alignment in the human entorhinal cortex?
No, it's just that the actual firing pattern itself is a regular triangular grid. And so it has orientational symmetry of 60 degrees. And so that's why every 60 degrees of rotation, you should see a similar, if you're looking ahead, you're running in a straight line ahead, then you should see a similar pattern of firing fields of all the different grid cells in front of you, because it looks the same every time you go through 60 degrees. That's because it's a regular triangular grid.
But that would mean any multiple of 60 should also work.
Yes. Yes, it does. I mean, the control experiments in that paper was looking at things that were not multiples of 60. 30 also should not work, because that, in fact, should be the optimal disambiguating.
Right.
And that's what we compared, sort of 30 versus 60. But we also looked for, instead of 6-fold rotational symmetry, 7-fold, 5-fold, 4-fold, 8-fold. And we did not see that. So that was the control for our method.
Right. So why do you think they're triangular?
Well, actually, that's an interesting point. So if you were an engineer, which you probably are, I don't know.
I'm a psychologist.
You're a psychologist. Okay. Well, if you were an engineer, then you might design some kind of navigation system or something to provide a metric on an X and Y right angular axes. So why do we have sort of axes that it's more like triangular axes with a 60 degrees instead of 90 degrees? And one reason you can see from Dead Reckoning. So in sailors that were navigating, they would try to have multiple lines that they were integrating across. And the reason why 3 is better than 2, because if I'm estimating my displacement in X and in Y, it gives me a location. But if I'm doing it on 3 axes, then any pair of them will give me a location. And I can check for my error by comparing it with any other pair. And so by having more than 2 axes, it's redundant, but the redundancy is useful because it tells you when you're accumulating error.
Right. Well, an alternative is that a triangle is the optimal way to cover a sphere, which would be interesting for grid cells, because that means it's sort of a never-ending, right, attractor system.
Well, you could. So it's close packed, and so you might think that you have 2D space represented on a torus, in fact, and you have close packed representation on that surface, and indeed, hexagonal close packing is likely. Is another good reason for why that you might end up with grid cells.
That's right.
There are models of how grid cells could be formed, say from place cells in terms of compression or whatever, and you would end up with hexagonal close packing.
That's right.
Yeah. Although in some cases, if you do the PCA, certainly in rectangular environments, you end up with 90-degree grids also. So
in our models, it's a twisted torus essentially with triangular packing, because that's optimal.
Yeah.
Okay. But so the other thing that you showed towards the end of your talk was actually an interesting effect, that if you have connected rooms but not continuous rooms, and you measure the grid cell response along these rooms, that they actually are sort of adjusting themselves with time, right? So how large can these adjustments be? Right. So what kind of shifts can you observe? Because it seemed to look like if these are connected rooms, they converge onto still a consistent mapping, right? A consistent coverage of then these 2 rooms as if it is one.
So what was the question?
Well, what kind of modulation can you expect to see there?
Well, so Francis Carpenter, who did this experiment with Caswell Berry, he recorded for up to 21 days of experience of the rats walking between these two rooms along the corridor. And after that long length of time, the grid-like patterns were becoming, in the two adjacent boxes, coming more like a global grid that covered both of them, but still hadn't got the whole way there. And so I guess what you could expect with experience, would be adjustments that can cover up to half a grid wavelength so that the two grids can come into alignment, whatever their starting phases are. It's interesting, in that experiment we had perceptually identical, as far as we could make it, boxes, so that the orientations of the grids would be aligned. I don't know if it might be much harder for the system with misaligned grids into two rooms to bring them into alignment. Although experiments by Jeff Taube's group has shown the equivalent thing in head direction cells, that if you have two different boxes in which head direction cells have different directional tuning, and you join them with a corridor that the rat can walk through, then eventually they line up and become consistent. And I would see these all as part of the same process of trying to build some kind of representation of space which is consistent to path integration between the different bits of space.
And do you see that as an adjustment driven by, let's say, CA1? Or is it extra hippocampal?
No, well, I would see in that case, because it's the path integration by walking backwards and forwards, which is presumably driving the reorganization, because that's what tells you the relative locations in the two boxes. So it's probably the grid cells trying to, if you like, align according to their movement-related inputs that drives the remapping, the slow remapping that you see in CA1 in this kind of situation, where place cells that are originally firing identically in the two boxes begin to slowly disambiguate the two boxes.
But there's still some monitoring process that can then check whether the grid cell response becomes more or less regular at some periodicity, right? Otherwise you won't get the correct alignment, because in terms of phase and spacing and orientation, it has to be aligned.
Yes, I'm not sure there's anything checking. It's more like simultaneous localization and mapping. So in robots, you come into a new environment, you try and you've got movement detectors, you've got sensory inputs, you try and build a map that's consistent with everything. And I think in this situation, perceptually identical, the perceptual feed forward, boundary vector cells and so on to place cells was dominating and making identical replications of patterns in both boxes. But eventually it manages to do something like SLAM and make a representation which is consistent with the path integration.
Okay.
But I would see it as, you can't put one before the other. It's trying to come to a compromise of both, and there's nothing checking.
Okay. But the plasticity sits in the entorhinal cortex driving this, or in the CA1 or CA3?
Well, there must be plasticity. Yes, I'm not sure actually. What I was going to say was there must be plasticity between the place cells and the grid cells, because the alignment must be. But I'm not sure now. Actually, it may be that the place cells are trying to anchor the grids to the environment, and the grid cells are providing path integration input to the place cells. It may be that doesn't require plasticity, and that the plasticity happens as the grids, if you like, pay more attention to their movement-related inputs than their sensory-driven inputs from place cells, and become more path integration-driven. And that, as a consequence, changes the place cells to become more consistent with that pattern.
Exactly.
And that actually didn't require plasticity between the place cells and the grid cells.
Yeah. On top of that...
But on top of that, then you must have plasticity with the two kinds of input. So I've posited the movement-related input that we think is driving the grid cells, and the environmental sensory input, which I think is driving the place cells. There must be plasticity in both of those inputs, because in the end, a place cell will, in one of the environments, will be firing differently relative to its sensory inputs.
Right.
And initially, the grid cells will be firing inconsistently with path integration because they're driven by the sensory inputs. So the plasticity in that model would be not between the place cells and grid cells, but between the place cells and the sensory input, and the grid cells and the path integration input.
But also because this emerges relatively slowly, right? It takes a long time to get this alignment. So how many hours does the animal spend between these two?
Well, that would be a couple of hours a day for many, many days.
Exactly.
And so one valid question is, if you made the task depend on knowing where you were in each box separately, maybe it would happen faster. Who knows? That might be worth trying because it's a horribly long experiment to run in its current form.
Exactly.
But we don't know.
Okay, great. But so we talked earlier about the concept cells, and in some sense, it's now the hippocampus is almost making Jennifer Aniston more famous than she was ever before. But we also have a new concept cell, which is now representing the concept cell of Jennifer Aniston. So if we start to think about generalizing away from spatial cognition and just behavior in space, and we start to think about other domains in which you can use these capabilities of the hippocampus, we still would need some sort of path integrator that drives that system. So imagine we're going to apply this now to, let's say, a complex cognitive task, and we're going to use the computational capabilities of the hippocampus. What would then stand in for its path integrator if I'm not moving in physical space anymore?
Well, I think as we discussed with this stretchy bird experiment and the grid cells, if you have a nice grid cell representation, it's very powerful and can represent the 2D vector relationships between any 2 points in a very large space. This could be very useful for mapping all sorts of things. Concepts, for example, where the vectorial relationship would be on vectors of neck length or leg length, or who knows how much I like them versus, how much they wear expensive clothes or anything. But it would be a powerful system for representing whatever that conceptual space is. And there, although path integration was probably useful for wiring up these grid cells in the spatial situation, if you have that representation somehow in this non-spatial situation, and maybe it's developed as a PCA of these non-spatial firing patterns, or maybe it's somehow the conceptual problem is mapped onto space implicitly. Either way, you then got a very powerful system for understanding relationships between different concepts.
Okay. So Neil, to finish up, okay, you come from physics, went to theory, and then to experiments. Now you combine this in animals and humans. And you also have been part of some really great discoveries in this field, and which also was recognized recently by being elected to the as a fellow of the Royal Society, which is a big compliment to your work. So congratulations. But the question now is, so given that experience, what would you see as Neil's law in the study of the brain?
I don't think there's any such thing as Neil's law. I do think that if you're trying to model something complicated like cognition, then at the level of neurons, you need to start with the simplest possible model because we have so little idea about how neurons do represent things like cognition. Obviously, in the spatial domain, we have some great clues from all of this work that we've heard about. And so I tried to always use the simplest model. And also it might not be a mathematically tractable model, although it would be great if it was, but it doesn't have to be. It's not clear that the brain is going to be mathematically tractable. And it needs to make experimental predictions, and indeed experiments have to have something to say to theory. They each only exist with the other in some useful sense. If you do an experiment, it doesn't impact any theories. What was the point? If you have a theory that doesn't impact any experiments, maybe it'll be useful one day, but it's not that useful right now.
But wait, the law of defining laws is they must fit on a T-shirt. So what's the law? Keep it simple.
Well, I think there's a somebody famous said something like, if I could capture my contribution, maybe it was Feynman in one sentence, it wouldn't be much of a contribution.
Okay.
I don't think that's true. I think there are some great discoveries which can be captured on a T-shirt, but I'm not sure.
I thought you were saying it. I thought you were saying keep it simple.
Yeah, okay.
Right. That's what I got.
That would be fine.
Keep
it simple. Okay. Keep it grounded in experiment.
Okay, good. Great.
It's not a very exciting T-shirt.
Well, it depends what else you put on it, but or who's wearing it. Maybe Jennifer Aniston. But look, 5 years from now, I'm going to smuggle myself into the UK because by then, after Brexit and the whole disaster that goes along, I will not be allowed to enter anymore in any legal way. And I'm going to come to your lab because by then you'll still be there. And I'm going to check whether a specific prediction you made today was actually verified or falsified. So what's the one prediction you really would like to see tested in that 5-year frame?
Well, there's a few. I mean, I didn't really talk about it because there wasn't time and it seemed a bit complicated, but I really would like to know if temporal coding and theta phase precession has something to do with path integration. And it may take nearer to 10 years, but I hope that at one point we'll be able to image grid cells and their dendritic inputs to work out what it is that's making them fire in a grid-like pattern. And there's a clear prediction that you should see oscillations of different frequencies in these different dendrites, and I would like to see that. Mm-hmm. At the more cognitive level, there's some applications to what happens in post-traumatic stress disorder in terms of different forms of memory supporting imagery or impacting them on them in different ways. And it would be nice to see if some of those predictions have come out, that this model of imagery had some clinical relevance.
Mm-hmm. All right, great. Neil Burgess, thank you very much for this conversation.
Thank you.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.